These are the lecture notes for Chapter 3 and its slide set B, or Part B of the slideshow. And we are still talking about transport across the plasma membrane. Excuse me. Here we go. Okay, so we already covered passive membrane transport, and the examples of that were simple diffusion, facilitated diffusion, and osmosis. So now we're going to cover active transport. And there are two major types of active transport. There's just active transport. That's all it's called, is active transport. And then there is vesicular transport, which is bulk transport. That's the transport of a large amount of material across the plasma membrane. Way too large for any, way too large for any carrier or channel protein to transport, and definitely too large to pass just directly across the phospholipid bilayer. So both types of active transport require ATP. They require ATP because they require energy. Okay. And another thing is that it's not on here, but down here at the bottom. So one thing is that active transport always requires energy, and the other thing is that the solutes are moving against their concentration gradient. In passive transport, they were moving down their concentration gradient, or with their concentration gradient. They were moving from high concentration to low concentration. But now we're moving them from their low concentration to their high concentration. And this requires energy. It doesn't happen passively. It has to have energy in order to cause it to happen. So active transport is going to require carrier proteins, which are often, a lot of times, they're called pumps. Like, for example, a sodium ion pump or sodium potassium pump. We'll talk about that. And sometimes they transport more than one substance. If they transport two different substances in the same direction, we call them symporters. And if they transport two substances in opposite directions, like one into and one out of the cell, we call them antiporters. There are two types of regular active transport. Before we get to vesicular transport, we're just going to talk about active transport in general. And one is called primary active transport. The energy for primary active transport comes directly from ATP breakdown or ATP hydrolysis. And then there's secondary active transport. So secondary active transport uses primary active transport to create a concentration gradient, and then the energy is obtained from that concentration gradient. And a lot of times it's easier to understand when you see examples, illustrated for you. So in primary active transport, the energy for active transport comes directly from ATP breakdown. And it causes solutes or ions bound to the protein, to the carrier protein, to be pumped across the membrane against their concentration gradient. So we have calcium pumps, we have hydrogen or proton pumps, and we have sodium potassium pumps, which is probably the most common and the one that you certainly need to understand and know that one. Sodium potassium pump is the most studied of all of these. Basically, the pump is an enzyme, and it's called sodium potassium ATPase. And it pumps sodium out and potassium into the cell. And we're going to talk more about how it works. I think these notes, if I were to read through them, they wouldn't make a lot of sense. And we're going to see some pictures in a minute. So the main thing is the sodium-potassium pump is essential for muscle and nerve function. So muscle contractions and nerve impulses. And now we're going to look at a picture so you can see this happening a little bit better. And I think I should blow it up. Let's see. Okay, so the sodium-potassium pump is actually this purple carrier protein, this purple pump right here. And what it does is the sodium in this case is the yellow molecule and the potassium is the green. So even though the concentration of sodium inside the cell is low and high outside, the pump still pumps 3 sodium ions outside the cell for every 2 potassium that it pumps in. So it says there are the first thing that happens is 3 cytoplasmic sodium ions bind to the pump protein. So you can see what's happening. 2 have already bound and then 1 is in the process of binding here. And so 3 sodium ions bind to the sodium-potassium pump. Sorry. And then it says the sodium binding promotes hydrolysis of ATP. So ATP breaks down. And this adds a phosphate to the pump or phosphorylates the pump, which is this purple. This is a phosphate here. And then the sodium, the shape of the pump changes and it releases sodium outside into the extracellular fluid. And now potassium is going to bind to the pump. Potassium from the extracellular fluid is going to bind to the pump, even though the concentration of potassium is already high inside the cell. That's why it's called a pump. And then it says potassium binding triggers the release of the phosphate, so that phosphate is now released from the pump, and that is going to change the shape of the pump and allow the potassium to move inside the cell, okay? So releasing the potassium ions inside the cell. So the bottom line is this, is that sodium is pumped out even though it's already got a high concentration outside the cell. The sodium-potassium pump continues to pump sodium out and continues to pump potassium into the cell even though the concentration is already high inside the cell. So the ions are being pumped against their concentration gradient and ATP is being used. And we will talk about the reason why this happens. Right now, I just want you to understand 3 sodium ions out, 2 potassium ions in. ATP energy is used, and this happens against the concentration gradients of the sodium and the potassium. Now, this is a very good animation that you really should watch on the resting membrane potential, because you will need to understand membrane potential or membrane voltage when we get to the muscle chapter, when we get to the nerve chapter. It's just going to keep on coming back. You may as well watch the animation now and start to get a grasp on the concepts. So this animation is slide 14. Now what we just saw was primary active transport. You had the binding of ATP and it directly caused a pump to pump ions against their concentration gradient into and out of the cell. Secondary active transport is going to be there's a little bit more steps to that. So the last note here, okay, so if we already know that sodium concentrations are high outside the cell, and that is because of the sodium-potassium pump and the primary active transport of the sodium-potassium pump, Then it's going to be real easy if you have leak channels, or if you have channels that will allow sodium to diffuse, it's going to diffuse back in because it's so high outside the cell. So when sodium diffuses back in, it can drag other molecules with it. Some sugars, like there's sodium glucose transporters, some amino acids and other ions, not just sodium, can be transported into cells by secondary active transport. So here's what we're looking at. We've got the primary active transport is coming from the sodium-potassium pump, keeping the concentration of sodium high outside the cell. So that's where the primary energy comes from. And the secondary part comes in because we have these other pumps or these other proteins, carrier proteins, in the plasma membrane that are going to use this concentration gradient that has been generated where sodium is high outside and low inside. So the secondary active transport is going to allow for the diffusion, passive transport of sodium and glucose at the same time. So it's using the energy from the concentration gradient created, by sodium. Since the sodium is going to diffuse in, it's going to take the glucose with it. And and that is the secondary active transport part. Now we're going to talk about vesicular transport, which is the transport of large material, either large particles, macromolecules, like proteins, and sometimes large amounts of solutions or fluids, and they're transported in sacs called vesicles. Now this still requires ATP, but the main thing is that the substance being transported doesn't have anything to do with a concentration gradient. It's just transported by a vesicle because it is so large. Okay, so if the substance moves into the cell, we call it endocytosis. There are three different types. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. And out of the cell is called exocytosis. So we'll look at endocytosis first. All right, and this is a picture that shows how endocytosis works. In fact, this is actually showing receptor-mediated endocytosis because the vesicle that's forming is coated with a protein. A lot of times that protein is called clathrin. And so, all right, so what's happening is the plasma membrane is forming an indentation. It's like it's forming this pocket. And the substance that's being brought in is collecting in that pocket. And eventually the plasma membrane will fuse and bring the whole entire substance into the cell, and it will close up. And that forms the vesicle. This vesicle is coated with a protein called clathrin. And that vesicle will release the clathrin a lot of times, and the clathrin can be recycled. But eventually that vesicle is going to fuse with, if you remember from when we talked about cellular organelles, the lysosome has digestive enzymes. The lysosome is actually a vesicle. It's actually enclosed with a vesicle itself. So you have the fusion of two vesicles. One contains the material that was brought into the cell, and the other vesicle is the lysosome. So once they fuse, those digestive enzymes in the lysosome will break down whatever this substance is. And a lot of times the next thing that will happen is all the waste will leave the cell by exocytosis, which is what you're seeing here, which is what you're seeing happen here. Then the vesicle will fuse with the plasma membrane and then push the material out of the cell. And this is all the steps that I just went over.